My dear viewers, welcome to Biology at Priyojit. In today's class, we explored the fascinating world of mitochondria, the powerhouse of the cell, and one of the most vital organelles in eukaryotic life. We will learn about the detailed structure of mitochondria, including the outer membrane, inner membrane, and cristae, intermembrane space, and matrix, and how each component is perfectly designed for efficient energy production. You will also uncover the key functions of mitochondria, from ATP synthesis and cellular respiration to roles in apoptosis, heat production, and metabolic regulation. This class also explains the endosymbiotic theory, revealing how mitochondria originated from ancient aerobic bacteria and enables permanent residence inside eukaryotic cells. By the end of this class, you will understand how mitochondria are built, how they function, and how they are evolutionarily significant, making this topic essential for undergraduate and postgraduate level studies. Introduction. Mitochondria is basically the double membrane bound cell organelles present in aerobic eukaryotic cells. They utilize oxygen for energy extraction and take part in Krebs cycle and oxidation oxidative phosphorylation. They are basically the powerhouses of the cell because they are the main sites of ATP production during aerobic respiration. Discovery. Firstly, mitochondria was observed by Collier in 1850, but named mitochondria was performed by Benda in 1897. Greek mitos means thread and chondrions mean granule. Mitochondria can be stained by Janus green and seen under light microscope, and this ultrastructure is studied under electron microscope. Number, position, and shape of mitochondria. Number of mitochondria fundamentally depends on the energy requirement of the cell. Its position varies according to its function. Around spindle fibers during cell division, at the base of cilia and flagella for movement. In muscle fibers, mitochondria are arranged in rows between contractile elements. Usually mitochondria are cylindrical, but they are highly dynamic. They can change their shape. They can fuse or divide by means of fission. Size. Mitochondria's length is 1 to 4 micron and diameter is approximately 0.5 micron. Structure of mitochondria. Mitochondria has two membranes, outer membrane and inner membrane, and two chambers, outer chamber and inner chamber, which is also known as matrix. Outer mitochondrial membrane, it is smooth and lipid rich. It contains porine proteins permeable to small metabolites. They contain enzymes related to lipid synthesis and tryptophan metabolism. Lower protein content compared to inner membrane. So outer membrane has lower protein compared to inner membrane. Inner mitochondrial membrane is selectively permeable and allows passage of only specific molecules like ATP, NAD, and coenzyme A. Inner mitochondrial membrane is very much protein rich, that is approximately 70 to 75%. It is rich in cardiolipin, making it impermeable to ions. It helps in regulate calcium 2 plus concentration in cytosol. Inner mitochondrial membrane forms inward force called cristae. It increases surface area for respiration, usually arranged at right angles to the long axis. We have already mentioned in our class. If more cristae indicates higher respiratory activity of mitochondria. Mitochondrial inner membrane also contains elementary particles or F0, F1 particles or oxysomes. Its number varies from 10 to the 4 to 10 to the 5 per mitochondria. Its structure, it has a head, that is F1, responsible for ATP synthesis. There is a stalk and a base is F0, which is act as a proton channel. So the proton movement through F0 drives ATP formation within mitochondria. So inner enzymes of electron transport chain etc. are located within the inner mitochondrial membrane, and there is an addition sites points where inner and mitochondrial membrane touch, which will help in the transport of materials within mitochondria. This is the image showing the structural features of mitochondria. Mitochondrial outer chamber, also known as intermembrane space, is the space between outer and inner membrane. Its width is approximately 60 to 100 angstrom. It contains water soluble proteins. Inner mitochondrial chamber is also known as mitochondrial matrix. There is a central cavity of mitochondria. It contains enzymes of Krebs cycle except succinate dehydrogenase, enzymes of amino acid synthesis and fatty acid metabolism. A mitochondrial inner membrane also contains circular and linear naked DNA molecules. These are the DNA molecules within mitochondrial matrix, RNA and ribosomes, which are 55S to 70S in nature, that is prokaryote type, and calcium phosphate and manganese granules. Presence of DNA within mitochondrial matrix makes mitochondria semi-autonomous in nature. So the semi-autonomous nature of mitochondria show partial independence because they possess their own DNA. Mitochondrial DNA replicates independently. They synthesize mRNA, tRNA, and rRNA. They have their own ribosomes. They synthesize some structural proteins and enzymes also. They grow internally. This is the image showing their mitochondrial division. So the mitochondria arise by binary fission. This is the image showing the binary fission of mitochondria, and this is the micrograph, electron micrograph of the process. So however, mitochondria are not fully autonomous because many proteins are encoded by also nuclear DNA, and their structure and function depend on nucleus and cytoplasm. So what is the endosymbiotic theory? This theory proposes that mitochondria evolved from free-living aerobic bacteria, and these bacteria entered primitive eukaryotic cells and lived as endosymbionts. Historical background: in 1883, firstly Andreas Simper first suggested symbiotic origin of organelles, and in 1905 Konstantin Meresinski proposed symbiogenesis, who suggested plastids originated from endosymbionts. In 1926, Wallin and Mechonsky applied symbiogenesis concept to mitochondria, and in 1981, Dr. Lynn Margulis gave the strong experimental evidence, and his idea was published in Symbiosis in Cell Evolution. Evidence supporting endosymbiotic theory: mitochondria have basically circular DNA like bacteria, and they possess 55S ribosomes, which are similar to prokaryotes, and it divide by binary fission. They resemble bacteria in size and shape. Their inner membrane contains cardiolipin, a bacterial lipid. Porine proteins present in both bacterial membrane and mitochondrial outer membrane. Their own protein synthesizing machinery they have. They are sensitive to antibiotics that inhibit bacterial protein synthesis. So the conclusion: mitochondria originate from aerobic bacteria, and the chloroplast evolved from cyanobacteria, we know, and the endosymbiosis played a major role in eukaryotic evolution. So the mitochondria, you are a powerhouse. So thank you, my dear students, for joining today's class. We explored the structure and functions of mitochondria and understood why this truly called the powerhouse of the cell. From the double membrane organization and cristae to their vital role in ATP production, metabolism, and apoptosis, mitochondria are essential for cellular life. We also discussed the endosymbiotic theory, learning how mitochondria originated from ancient aerobic bacteria as an idea strongly supported by their DNA, ribosomes, and independent replication process. I hope this class helped you clearly connect mitochondrial structure and function and evolutionary significance. Stay curious and keep learning. See you in the next class.